Okay, let's start. Good morning, everyone. Welcome to this seminar in Ecology and Evolution of Montpellier. For those of you who are online, as usual, you are in a webinar mode, which means that you cannot open your mic and your camera. If you have any technical issue, you can report using the chat. And if you have questions to our speaker for the question session, you can use the Q&A tab in Zoom. And so with this, I give the floor to the enlightened scientist who will introduce today's speaker.
Good morning, everybody. Well, we are very pleased to welcome Julie Louvrier. So Julie is ecological modeler. She did her PhD here at the CEP with Olivier Jimenez on a species distribution model on large carnivores, with a case study on wolves in France. And then she did a postdoc in Germany, in Berlin, at the Leibniz Institute, the Institute of Zoology and Wildlife Research, with Stephanie Kramer-Schat, and yeah, I'm sorry if I pronounce it wrong, and on the urban ecology and the species distribution model. It also used a hierarchical Bayesian model and occupancy model. And then you continue with Victoria Rachnok, also in Berlin, the same institute, more on the demographic resilience. And today she's going to be talking about, well, now you are back to in Montpellier, and she has a junior chair professor at the CNR-IRSA, working on the wildlife ecology, epidemiological model, and like how movement patterns of animals influence transmission of infectious disease. Maybe you can talk about it a little bit later at the end. And today she's going to be talking about a case study, a simulation study, occupancy model with using a co-occurrence data.
Thank you very much. Hey, everybody. Is that okay? Can you see here? Or yeah? Okay. So hi, everybody. Thank you for coming to the seminar. It's an honor to be here presenting my work and to be back to Montpellier and to be back to the CEF where it all started for me. I did my master, and even before I did my gap year, my CESUR in the CEF. So it feels a little bit like coming back home. So today I will talk about a simulation study that I conducted with Olivier Jimenez, Melissa Cudino, and Josefa Vergara, who was a student who worked on those models when she was with me in Berlin. So you'll see we basically did a small simulation study. I am happy to present here now because it is in the review process and I need to get this work done at some point. So I thought it was a good opportunity to bring it back, present it to you guys, share the ideas if you guys have some ideas about this project and this simulation study, and also talk a little bit about what I'm going to do in my Chair de Prof Junior, so in my next job. So before I start, please enjoy those nice pictures of animals, because this is the only cute picture of animals that you will see in this presentation. I click. Do I click? I click. Yeah. So to get back into the topic of species interactions can be defined in a lot of ways, depending on what we're measuring, depending on what we're looking at. It can be a lot of different things. Here, I'm talking about interspecific interactions, such as predation, competition, or mutualism. Those are big factors when we talk about the ecological niche segregation, either temporal or spatial. And there's a big question, which is how can we measure species interaction? It's a very complex question, and I guess you all have an idea about this. And in my case, we investigate co-occurrence. So the presence or absence of species with the presence and absence of another species. Here we're talking about two species, but it can be multiple species. And in that case, we're talking about spatial co-occurrence, not temporal co-occurrence, but spatial co-occurrence. The tools that we use, those are tools that have become very common, which is occupancy models, which I think most of you are familiar with. They have been developed they have been developed in 2002 by Darryl McKenzie and collaborators. But ever since, they became more and more flexible tools to answer a lot of ecological questions. That can be a lot of different applications, but in our case, we'll talk about species interactions. So when I talk about species interactions, I actually talk about co-occurrence. So I'm sorry if you guys were thinking about something else, we'll talk about co-occurrence in that case. So occupancy models, what is occupancy? Occupancy is the proportion of an area that is occupied by a species. So you take an area, you divide it, and you look at the presence of the species. It depends on a lot, it relies on a lot of assumptions, such as the closure assumption, and all of this. I am happy to discuss the assumptions after, if you guys want to know a little bit more. And we talk about the probability that a species occupies a site at a given time. So this obviously relies on how we're going to define what is a site, on the biology of the species, if the species is mobile, is highly mobile, is migrating or not. So there's a lot of assumptions behind it, but here we're simplifying the story a lot. And obviously it is the function of biotic and abiotic factors, such as presence of prey or climate or altitude, or depending on the biology of the species once again. So what we have different sites. Here in our case, we defined 6 sites, and this is the study area. On each of the sites, we're going to survey and repeat the surveys, go on the sites and try to see whether or not there are traces of the species, present signs, feces, tracks in the snow, camera trap detections. It can be any types of present signs. And then you get those 0 and those 1s. So 0, 0, 0, the species was not detected during the 4 occasions. And once the species was detected. It is very important to account for the sites that were not sampled. And this is a whole different topic as well that I invite you to look for, which is what is my sampling bias? In which site did we go or did we not go? Because then you might bias your results. But also how many people went on the field, for how long were my camera trap active. Like, this is very important to account for. And for instance, reality can be quite different from what we see, obviously. And so, for instance, if we look at this site, I think I highlighted it. No, I did not. So here, if we look at this site, although we went there 4 times, the species is present. And we're talking about imperfect detectability. So, which is the fact that even though we go there, we sample the site, it's likely, there's a probability that you might not detect it. So here, once we know a little bit what is occupancy models and why it is nice to use those models, because they allow us to get into the layer, the ecological layer, not the observed one, but we look at the ecological layer because we account for this observation process. In our study, we looked at the paper, at the model developed by Rota and collaborators in 2016. And I invite you, if you're interested in these models, to read this paper that was published in Methods in Ecologie-Evolution, titled A Multi-Species Occupancy Model for 2 or More Interacting Species. So this paper, this model was developed in 2016 and started to be developed and started to be used by a lot of ecologists and in different frameworks. But until now, there was no simulation study or assessment of how good this model is actually. It is widely used, but we don't know if it actually, maybe it estimates biased estimates, maybe we don't really know. So in our case, we thought, okay, let's do a little simulation study and fit this model, see how good it does. Before doing so here again, a little cute animal to illustrate what we're going to do. So in that case, we have 2 species, species A, which is a lynx, and we have species B, which is a chamois. And so what we're modeling in those cases is 4 possible scenarios. We have sites with both species that are present, and we have the probability that the species B, so the subordinate species, it can be here in our case, we're talking about the prey, is present, given that the predator is present. On the other hand, we have the probability that the predator is present, given that the species is also present. Capital letter means present, small letter means absent. We have the second case when the predator is present, but the prey species is absent. So we have small b, capital A, small b. And we have the case where the prey is present and the predator is absent. Capital B, small a. So every time I do this presentation, at the end, I start to get tired in the probability that the species is present given. So please forgive me if I start to stutter on those, because it's every time it's kind of a little exercise of pronunciation. And we obviously have the last case, which is both species are absent. So it is possible to measure those four, to estimate those four probabilities. So psi is usually the symbol that is used in occupancy framework to define the probability of occupancy. Again, are they really interacting? I would not go that far, because interaction is actually a very complicated question. Are they really interacting? I mean, given that lynx predate on chamois, I would say yes. But are we really measuring this? I would say we're measuring a proxy. So we're actually looking at this proxy for interaction. We're looking at the conditional probability of occupancy, given that the other species is present or absent. So we're really looking at whether this probability, C, capital B, capital A, is different from psi, capital B, capital A, and vice versa. So, well, that's a lot of typos on the title, I'm sorry. So how do we quantify the statistical associations? So we all know that we need sufficient sample size of locations. How many? We always go with a rule of thumbs or what we have, given the logistics that we have as well, because sometimes it's not possible to sample as many as we want because of logistics, because of financial or people, maybe it's complicated. We need to have sites with and without the species of interest, and we need to have repetitions. How many? So all of those are questions, and when we read the studies, we can see that there's a bit of a rule of thumb and where people think it might be sufficient and all this, and we wanted to just assess what would be the golden rule for if one wants to estimate those parameters. So we created 7 interaction scenarios from low to high intensity of interaction or avoidance. So we're talking about avoidance in that case because we're looking at the probability of presence of the species of the prey species, so the subordinate species, given the presence and absence of the predator. So here on top, 0 is the name of the scenario because there's no difference. So here, both probabilities are the same. And then we increased, so psi A, so the probability of presence of the predator is always the same. We decided to keep it as it is, so then it's not getting too complicated. And then we can see here that the more we are going to increase in our scenarios, the lower the probability that the prey is present, given the predator is present, is going to lower. And we keep this one also stable. So here we're going lower, so 0.4 here, and then 0.3, 0.2, 0.1. And we thought, okay, let's test one extra, which is kind of like really something that might be a bit more towards absence, where here we put the probability of presence given the predator is absent to 0.8. So 0, 1, 2, 3, 4, 5, 6, the more we go in those numbers, the higher the avoidance. So we did our simulation study with 3, 5, and 10 repetitions. So that's the number of time people are going to come on the sites and survey the species. in the little drawing that I showed, it was the number of repetition was 4. We tested for 30, 50, and 100 sites. And then we added, because we had some recent reviews that asked us to do 150, 200, and 250. So it's not on this slide, but it is in the results. We worked on the probability that at least the species will be detected once during the repeated surveys, and we set it to 0.6 for the predator, 0.8 for the prey. Why, you may ask? We just wanted to test really good detection probability. If you guys have worked in occupancy models, the detection probabilities can be very low. So for the wolf, when I was working on the wolf models, the detection probability is around 3%. So here, 60% is amazing. It's really great. 80%, easy. So we wanted to test kind of a best case scenario when it comes to detection probability. We did 100 repetitions for each scenario. We simulated each of the scenarios, fitted the ROTA model, but also a model without interaction. So in the ROTA model, one can set whether or not there's an interaction. So if there's no interaction, it's just 2 occupancy models that are fit in parallel. We measured the mean squared error, which is the mean of the difference between the true parameter and the estimated to the square. Measured the relative bias. So the MSE allows us to measure how precise our estimates are around the true value. So it gives us the precision. We measured the bias, which gives us how far are we from the estimate and the true value. So we had the true value. So that's actually why we're doing this. And the coefficient of variation, which is the standard error divided by the mean times 100, which gives us an idea about how dispersed the points are around their own mean. We also calculated the difference of AIC between the 2 models, the one with interaction and the one without interaction. So actually, AIC without minus AIC with, as a way to understand when the model is actually performing better than a null model. So, in terms of results, we will take this slowly but surely. So this is the mean squared error that we're seeing here. And what we're looking at in the axis is the number of sites. So from 30 to 250. And here we have the mean squared error for psi A, the probability of presence of occupancy of the predator. Psi capital B, capital A, so the prey is present and the predator is present. And here the prey is absent and the predator is present. The avoidance level is represented by the colors. So 0, it's when there's no interaction. And 6 is the maximum. If you remember, this is the one that was put to the extreme. So yellow is avoidance at the maximum. 0 is there's no avoidance. What we're seeing, first, the number of sites helps with the precision. The more sites we have, the more precise the estimates. Good, so far, this is good. It's kind of what we expected. So, but it's always good to verify this and to check that. So this is good for the 3 parameters. Second, what we're seeing is that the precision of one parameter, the one of the when the predator is absent, is more biased than the one when the predator is present. And this is actually also led by the fact that we have the interaction, the avoidance level. We can see that the more we go into a strong interaction, the more the signal is clear, the more we're getting into precise estimates. And this is for the 3 parameters. So far, so good. This is awesome. What does it mean? The interaction scenarios influence the precision, as we saw. The repetitions, so this I didn't show, and it's in the appendix. I can show the graph after. They're ready. It's everything done and ready to be put. The number of repetitions, they don't seem to affect the parameter estimation. So here we have the number of sites before, but same, we tested with the number of repetition because we have high probability of detection. of course, it's not going to influence the parameters estimate. So if we were to have lower detection probability, then we expect the number of repetition to increase the precision. But we thought it was adding a bit of complexity to the message, so we stayed to this. The number of sites seem to have a bigger influence on the precision of estimates, as we saw. MC capital B capital A is more precise than psi capital B small a. Now, the bias. So what do we see here? The number of sites also influence the bias. So the more sites we have, the more we sample in space, the less bias our estimates are. We set the limit of 0.05. It's a relative bias, which means that when we look at the limit of 5%, if it's below 5%, it means that it's actually not so biased. So the model is doing quite good in terms of bias, except for one parameter, which is C capital B small a. And we see here that there's a huge also variation in the values of bias. When it comes to the avoidance level, I don't know about you, but I don't see really a pattern in the avoidance level. So all we can say, what does it mean? The interaction scenarios, they don't influence the bias, maybe a little bit, but I don't see it. Repetitions don't seem to affect the parameter estimation neither. So this is, I will show you after. The number of sites seem to have the bigger influence on the precision of estimations and the bias. And C capital B, psi capital B capital A is less biased than psi capital B small a, but good values of bias, except for this last parameter on the right panel. Coefficient of variation, I will go a little bit now. You're starting to understand those panels. It's always the same representation. It's the same color code. It makes it easier to understand. Now the coefficient of variation, what we have is actually something different, which is that now it's this parameter that has the higher variability around their mean. We have an effect here, we can see clearly, an effect yellow to purple. So from 6 to 0, when we have low values of avoidance, we have low values of variation. I will show you why. I investigated a little bit. It's still not very clear, but I think I'm starting to have a key interpretation of this. So the interaction scenarios influence the coefficient of variation. The repetitions don't seem to affect the coefficient of variation. And the number of sites, they have the bigger influence on this coefficient. And now this time we have psi capital B capital A, which is bigger than psi capital B, just one B, small a. Now the delta AIC. So we looked at the difference of AIC, and amongst our 100 simulations, for each scenario, we looked how many times we had a delta AIC that is bigger than 2. 2, I know that there's a big discussion about the value of delta AIC. It can be 2, some people say 7, some people say 9. So of course, I am happy to discuss this, but if we take it more than 2, you will see that actually the conclusion is quite gloomy. Not gloomy, I mean, this is just a simulation scenario, so. But okay, so here in our case, what do we have here? Here in horizontal, we have the avoidance level, so no avoidance to high avoidance. And here we have the number of sites, so 30 sites, 50, 100, 150, 200, 150. And we have the number of temporal replicates here. Left panel is the proportion of delta AIC that are higher than 2. And on the right panel, we have the proportion of p-values. So we can also have an estimate of the p-value of the interaction parameter. Because in some studies, researchers look at the p-value, not at the AIC, so they look at the p-value of the interaction parameter. And they say if it's below 0.05, it is significant. I know also the whole conversation about p-values and 0.05, I know. So we can discuss this, happy to look at different values of 0.5. It is a threshold. As soon as we decide the threshold, I know we're making a lot of assumptions. So in our case, what do we look at here? The red line is once we have 75% of our scenarios for which the delta IC is bigger than 2. This is the scenarios, so this is the scenarios in which the model accounting for interaction performs better than the model that does not account for interactions. And in most of the cases, here we can see we are below 75%. All the other values, you see the proportion, it's between 0 and 0.75. The darker it gets, the closer we are to 0.75. When we look at the p-value, here we can see that this region is even smaller. So it means that, in conclusion, if one wants to have a significant interaction and make sure that there's a significant interaction, we need to be either have high sampling size, a high number, but also to be in a high avoidance scenarios. Otherwise, there are high chances of not detecting, although there is interaction, there are high chances of not detecting the interaction. Okay, so what does it mean? Only in high interaction scenarios does the model accounting for interaction perform better than the one not accounting for interaction. The number of sites helps. To have a model performing better, we recommend at least 150 sites. And something to keep in mind, the absence of evidence for an interaction does not necessarily imply that there's no interaction. We all know that the absence of proof is not the proof of absence. So we need to keep this in mind. But this means that every time we're looking at this model and the results, even though there's no proof of interaction, there might be interaction going on, but we don't know. It's hard to prove. We conducted a small literature study that used this model. So we actually, out of all the studies that we found actually 13 empirical studies that were actually reporting the parameters that we're interested in. In the empirical studies, we had 4 with less than 150 sites, which is 150 is a bit the number that we recommend. The avoidance strength went from, if you remember, there's low interaction, which are numbers between 1 to 3. We have Siberian ibex and the gray wolf by Salvatori and collaborators in 2021. The black bear and grizzly bear by Ladol and collaborators in 2018. And 8 is the strong interaction parameter, was found in the gray squirrel and the martens in an urban setting by Twinning and collaborators in 2021. So the avoidance parameters is also changing depending on the study, depending on the species that are studied, obviously. And the number of sites, the minimum that we found was 14 sites with a study by Said and collaborators in 2022 on snow leopard and I think it was ibex and blue sheep. And the maximum that we found was 1,427 sites by Parsons and collaborators in 2018, which was about, I think it's urban species, and I think it was coyotes. Yeah, and then the number of temporal replicates varies a lot. We have minimum 4 temporal replicates by Cass and collaborator 2020, and more than 20 temporal replicates by Ladol and collaborators in 2018. So what can we conclude? Well, the reality is that when one sets a study and wants to study species interaction, the reality is that it might be complicated to have the ideal number of 150 sites and the ideal repetition and the ideal detection probability. What can we conclude on our side? That results, when we have less than 150 sites, need to be taken with a pinch of salt. So that's the main message. And after this, we need to account for ecological complexity. We were not having environmental factors, the effect of environmental parameters. We were not having other species presence or absence. So life is much more complex than a simulation. But it is a starting point to actually give an idea of how good those models perform. So now I would like to thank you. I thank my collaborators, Olivier Jimenez, Mason Filino, and Josefa Vergara. I would like to organize also the organizer of the seminar for inviting me and everybody for your attention. And if you have a bit of time, I can talk about what I'm going to do in my new job position, because I'm going to look at species interactions based on camera traps, but this time with a link with domestic species and the effect of human activities on the overlap, the spatial overlap of domestic species and wildlife in the context of infectious disease transmission. So thank you very much.
Are there any questions?
Just a curiosity question, these co-occurrence models, are they used in, for instance, niche environmental niche modeling to predict joint presence and perhaps joint changes under changing environments and these types of things?
So I think for prediction aspect, no, not so much. But they're just for assessing whether or not there is a strong interactions between two species, not for the prediction aspect. I haven't seen
Because
that.
it would just involve putting a layer of environments.
Exactly, yeah, and then predicts, yeah. Yes?
I don't know if I got all the information from the graphs correctly.
Okay, yes.
But so it's my bad, but the 150 that you say, does that, oh, is that like if you know something prior, if you have prior information on the interaction, it's going to be more in this range like this, is that maybe something that can use the 150 or?
Yeah, so I mean, so if I understood your question, can we use prior information to test whether or not, if we know that we have a strong, and then look at this and think, okay, maybe we don't need 150 sites, but we need lower. Yeah, I think this is a concrete example if one knows already about the strength of the interaction in the maybe literature or previous study or something like that. Yeah, I think it's a good idea.
For example, the index and wolf, you know, you have an idea what it is, so you don't need 150, which could be difficult in nature. So.
Yeah, and I mean, yeah, definitely, because I think the strength of the interaction depends on the whether or not a predator is actually specialized on a prey, for instance, and I would assume that predator-prey interactions are actually stronger if the predator is specialized on a prey. So exactly, that's exactly the type. But for instance, interspecies like competition, it would be a bit more complicated. But to know exactly what is the strength of the interaction, yeah, I agree. Yes.
So there's no abundance here; it's just occurrence. You can imagine that competition or predation acts more on relative abundance than co-occurrence. Actually, is there a way to take abundance into account? Or maybe because abundance should influence detection probability. So
exactly,
including the detection probability in the models.
So there's a new model that was developed by Mason Fidino, actually, which is one of the collaborators. I think it was published in 2024, where he developed abundance-based interaction models. So it's actually a similar framework, but it goes deeper into the effect of abundance. Yeah, because here we're looking just at 0, 1, so we're losing a lot of information. Yeah. So yeah, I invite you to read the paper by Mason if you guys are interested in this. Yeah.
In protocol studies, you also have to decide whether to do more sites or fewer sites more often, or this kind of things. So what would you advocate for looking at interactions?
So if you're looking at interaction, would I look at more sites with less repetition or less sites with more repetition? I think it depends on the detectability of the species. If you have a high detectability, I would go for more sites and less repetition. If you have a lower detectability, I think I would focus the effort on less sites, but repeat it, because otherwise you might miss the information. And the first information is to detect the species. So I would go for that. That's just my 2 cents. Yeah.
Thank you for the talk. Is there any relationship also with the size of the sampling sites? Because I would guess, like, for example, if you have an interaction of predation, you would think that the two species are in the same region, but not maybe in a different scale, that it is a relationship about mutualism, like because they're kind of avoidant, but if you create a sampling site, they have to be in the same region.
Yes. Yeah, so this is all related to the closure assumption. So in occupancy studies, you have to define the size of your sites based on the biology of the species, because you have to assume that between your repetition, the status of the site, so whether the species is present or not, does not change. So the more you repeat, the species cannot migrate or cannot, go around because it has actually a territory that is bigger than the site. So then actually what you're seeing is not occupancy per se, it's the use of space. So it's a bit different. And then when you add two species, yes, you need to account for the biology of both species, because usually predators, they have much bigger territories than prey species or ungulates. So this is actually something to account for, so you need to encompass the presence of both species at once. So yeah, that's actually, one of the critical questions, which is how do we define a site? And how do we repeat? Because then depending on the biology, then you have yearlings, offsprings that are dispersing, and then it changes everything. So yeah, I agree with you. It's a complicated question, especially the more you have species involved, the more complicated it gets.
We have a question online from Christine. Christine, can you open your mic?
Can you hear me there?
Yeah, that's perfect.
Yes, thank you, Julie, for an interesting presentation. I wanted to hear more about what you're going to be doing. Are you going to be based at CEF? And more specifically, what do you anticipate to be like the big challenges of using camera traps as inputs for occupancy models?
Yeah, thank you very much for this question. So, because I'm right in the beginning of starting new projects, and I think this is something that is in my mind quite fresh. So I'm not at the CEF, I am at INRAE, and I will work on the link between wildlife ecology and domestic species presence. So the project that I have in mind, the first one that I'm going to set up is a camera trap study in Cambodia, where I have colleagues who have been working there for several years and study the impact of human disturbances on biodiversity communities. And so what I'm expecting, they have different sites on an urban gradient, so from very urban areas to almost pristine forests, and they're trying to assess whether or not the biodiversity is going to influence the emergence, emergence of infectious diseases. And so what I'm expecting, so I would like to look at species interaction in the sense of looking at wild species and domestic species, whether or not they are avoiding. My assumption is that wild species are avoiding domestic species and they're avoiding humans. And what I would like to see is detection of humans, so I can measure the activity of humans and their presence and see whether or not the presence of humans, indirect or direct, is going to make wild species have their spatial niche overlap. And so if you have a spatial overlap, then you have more chances of disease transmissions. So I don't know if that was answering your question, but this is a little bit what I'm going to look at in the next months, because October, the PhD will start and I will start advertising it. So if there's anybody interested in wildlife and doing fieldwork in Cambodia, and yeah, that's not the worst place, huh? And applying this type of models, looking at species interactions and also temporal interactions, because here I was talking about spatial interactions, but I also look at the use of the temporal niche, because the more you have humans play the role of hyperkeystone. So we're very active in the day. And so what we have noticed as well in my postdocs is that when we're active during the day, wild species are restraining their activities to even more nocturnal activities. So I'm kind of interested in looking at this, not only spatially, but also temporally, to see whether or not they will occupy the same temporal niche. That was a long answer, I'm sorry for that. Any other question?
Is there any question online?
Doesn't seem.
There's a question here, yeah?
Yes, I was wondering if you invest a lot of money and energy in building a solid co-occurrence model in, for example, a restricted number of sites, then you can use this model to improve the detectability of one species based on just the detection of the other one in the rest and also.
Yeah, so this is actually done in multi-species, so not only two, but multi-species kind models developed by Royal and collaborators. So it's another type of models, but they actually use the inference of the common species and the fact that, you know, whether or not they're detected, they're going to assume the probability of occupancy of the other species that are less detectable. Yeah, so it can be used in that way, but the more species you have, the more likely you are to infer the presence of a species that is less detectable.
I have a last question. Going back to your study, when you were talking about the closure assumption, because the WOTA model is a closed model, right?
Yeah.
Have you think of potentially do the simulation study for the open version of the model, and what would you expect in terms of bias?
I mean, if it's open, then it's going to be even more biased. I think we kept it very simple to start with the best case scenario in that sense of no environmental variation, closed population, and all of this. We are not thinking of doing an open population setting because I don't have the time anymore to work on it, but I think that would be very interesting to look at it. Yeah.
We need even more science and
I'm assuming we would need more sites and more repetitions and also like more
But what
information.
you could have on the colonization versus extinction, do you think it would be affecting more the occupancy or the dynamic parameters?
Yeah, I mean, you mean extension or probability of staying like extinction and colonization parameters? Yeah, I don't know. Yeah, I don't know. Yeah, happy to talk about it, to brainstorm over it and during lunch. Because I know you know those models as well, very good. Mm-hmm.
More questions? Okay, thank you very much.